Hey, my name is Sai. I work with the Biomedical Imaging Lab at the University of Louisville, and my research is focused on investigating color normalization and how it impacts kidney image segmentation. So, I'm running into some technical. Okay. So segmentation is when we are able to identify certain segments of an image. So we can see that this is different from just object detection, where a bounding box is used to delineate different parts of the image. So with segmentation, objects are assigned a pixel mask, so it's much more precise. There's different kinds of segmentation. So with semantic segmentation, the entire class is assigned one mask, where with instance segmentation, each instance of a class is assigned a different mask. So we can use deep learning for segmentation and actually train a model to recognize certain objects for us. This project deals with the segmentation applied to specifically medical data. So specific structures within tissue images or histopathological images can be recognized. And this is very important because oftentimes segmentation can be more accurate and efficient than pathologists. An example of a segmentation pipeline is illustrated here. So we have a whole slide image which depicts some sort of organ. These whole slide images are very large, and they're too large to deal with. So we can split them up into different patches or tiles. We extract features from those tiles, and so then we can determine which features are important to the object that the object in the image that we are trying to recognize. And then we go into the machine learning aspect of the pipeline. And since training a segmentation model relies on having some ground truth annotations, we're working with labeled data. So typically it's supervised learning. And then from here, we can obtain some sort of segmentation map. So one of the objectives of this research is to build a model capable of identifying glomerular functional tissue units in kidney tissue. And so these are structures in the kidney that are essential to filtering the blood. But the main objective of this research is to evaluate the impact of color normalization techniques on the accuracy of a deep learning model trained to recognize glomeruli from kidney tissue. And the reason that kidney data in particular was chosen was because kidney tissue is very, very heterogeneous and complex. So by being able to get a better understanding of how color normalization can impact segmentation performance with this kind of data, we can gauge its performance with other kinds of data. So color normalization is a very important pre-processing technique. When we have tissue, in order for it to be properly visualized, the tissue needs to be stained. But there are a lot of different staining procedures, and there isn't exactly one standardized technique for staining. This can result in data sets that have major discrepancies in their staining patterns, and that makes it more difficult for a deep learning model to train on that kind of data and maintain a good accuracy and to be reliable. So color normalization can help us alleviate that problem. With color normalization, the color pattern of a reference image is transferred to a source data set, and the result is a normalized data set that is generally more aligned in color. So the data set that we are working with includes 15 formalin-fixed, paraffin-embedded whole slide images, and this data came from the HubMap Hacking the Kidney dataset. These images were visualized with the PAS, or periodic acid shift stain procedure, and I decided to actually split each whole slide image into patches of 512 by 512 pixels. Also, this data came pre-annotated, so the location of the glomerular functional tissue units were given to me already. So going into the specifics of the color normalization techniques that we're working with, the Vahadane method uses stain decomposition. So the images are decomposed into non-negative and sparse stain density maps. And this is because stained histopathological images are sparse or non-negative in nature, since some regions either have thinly distributed stains, or some regions have no staining at all. And this method typically works well with staining methods that use multiple colors like H&E, which stands for hematoxylin and eosin. But it is compatible with periodic acid shift as well, which is what the data set that I'm working with is represented in. So once we have achieved a separated stain maps, they're then transformed to match the color space of a reference image. And then these different density maps are combined to form one composite normalized image. The Macenko method is very similar to the Vahadane, since it also leverages stain decomposition. But a unique aspect of this method is a step that specifically focuses on estimating the concentration of different stains. So based on the stain distribution of a reference image, the stains are transformed and then recomposed for, again, one single composite normalized image. So the Reinhardt technique is actually, it's a little different from the other two. The Vahadane and Macenko techniques were made specifically in the histopathological context, while the Reinhardt method sees itself in many other contexts. The Reinhardt method compares the mean and standard deviations of the color channels from the target image and then transforms the source image on a channel by channel basis to match the reference image. So with every single normalization method, what you'll notice is that the normalized image always retains its structure while closely emulating the pattern of the reference image, which is precisely the goal. So after normalization, it's important that we apply augmentative techniques in order to proliferate the training set, since the data set that we're working with is very limited in size. It's only around 2,600 images. So augmentative techniques improve the generalization of the model and can improve accuracy as well. The techniques that I used included random mirroring, random cropping, random jittering, random noise. It's important to mention that the train test split was also 90-10. So 90% of the training data was allocated for model training, while the other 10% was set aside for testing. So going into the deep learning model that I used the UNet model, and we can immediately notice the unique shape of the UNet model. It's U-shape. The UNet model is specialized in segmenting biomedical image data, and it's among the most prominently used models with this specific context. So previous works that have also segmented glomeruli from kidney tissue data have also relied heavily on the UNet model. So the first half of the UNet model is what we call the contracting path, and the goal of this path is to condense spatial information and extract features. The second half of this model is the expansive path, and this path uses something called transposed convolutions. So basically like upward convolutions to get the image back into its original resolution because the contracting path condenses that image in order to extract features. So the expansive path uses both spatial and feature information and culminates that into optimally a high quality output segmentation map. What really makes the UNet model unique, however, are the skip connections. And so the skip connections are essentially features that connect the non-adjacent layers. So basically the contracting and expansive paths to each other. So when information is passed through these successive layers, some of that information can be diluted and lost. But if we can preserve the rich details of the data and propagate it to the latter stages of the model, that's what we're doing with skip connections. Since we're working with histopathological data, this feature is especially useful since histopathological data is very rich and detailed. And just to conclude, the UNet model as a whole contributes to high quality and localized segmentations. So going deeper into the model that I developed, its specific parameters, I set the batch size at 8. So every time the model processes 8 images, the weights of the entire model are updated, and the model is trained for 100 epochs, meaning it passes the entire training data set 100 times before arriving at finalized weights. I use the Atom optimizer, which is responsible for updating the weights through backpropagation, which adjusts the model based on the results it's getting. I then use the dice loss as my loss function, and the dice loss is 1 minus something called the dice score. And the dice coefficient measures the overlap between the model's predicted mask and the ground truth, and the ground truth is just like the actual mask or the actual location. So the greater that intersection, the more indicative of a strong model performance. And as I said, the dice loss is simply just 1 minus the dice coefficient. So the first phase of testing actually involves gauging. I wanted to gauge the quality of the normalized data sets. So in order to do this, I decided to utilize an array of color metrics rather than just rely on one single metric. So these are the color metrics I used. I used the Pearson correlation coefficient and the functional similarity index measure, which both assign scores based on the strength or quality of an image compared to an original image. So in our case, this would be comparing the normalized image to the original non-normalized image. I also used the universal quality index to measure overall image quality of the normalized images. The second testing phase is where we actually evaluate the impact of color normalization on segmentation performance. So once the normalized data sets were fed into the UNet model, they would be evaluated and compared with the dice coefficient. So the reference image that you see at the top was actually chosen based off of its prior success. So several other people or teams that have worked with this HubMap hacking the kidney dataset have implemented color normalization techniques, and they have used that topmost image as the target or reference image. And we can see the normalized images here at the bottom. And I thought how different each normalized image was from each other. I found that pretty interesting. And we can also notice that from the original image, there's some dips in image quality because of that normalization process, which is completely expected. So before I go into the results of the color metric application, I wanted to go over these HSV plots. So I wanted to explore and visualize how color normalization actually impacted the nature of the data. These plots are a good visualization of color normalization due to the bimodal nature of histopathological images. So in order to actually achieve these graphs, I converted the typical RGB image into the HSV or hue saturation value file format. And then I extracted the hue and saturation channels and plotted them against each other. And we can see how dispersed the non-normalized data is and how much more concentrated every single normalized data set is. I also want to point out that the plots for the Vahadane and Macenko techniques look very similar. So the points, each point that you see on the graph, on each graph represents one image. So these points are congregated for the Macenko and Vahadane plots. So they're congregated in a pillar-like orientation, while the Reinhard plot depicts a much more circular cluster. So looking at the average values of the color metrics, we can see that the Vahadane algorithm generally boasts the highest average values, at least for the universal quality index and the functional similarity index metrics. I deducted that this could potentially mean that the Vahadane normalized dataset could be of higher quality and that could potentially lead to better segmentation results. So moving into the main results of this research, the segmentation results, I used K-folds as a cross-validation technique. So I split the data into 10 different folds. So there were 10 different testing data sets. And the corresponding table depicts the average die-score coefficient value of the 10 trials that were accommodated for each data set. And the main takeaway here is that every single normalized data set, regardless of the technique that was used, outperformed the segmentation performance of the original data set. Also, the Reinhardt algorithm narrowly outperformed Macenko to be the best performing technique. So just to conclude, of the 3 common color normalization techniques tested, all of them contributed to higher segmentation accuracies. The Reinhardt algorithm was the best performing. And another takeaway is the color metric results, which were intended to gauge dataset quality, did not necessarily correlate to the segmentation accuracies. Another key takeaway here is that color normalization can improve segmentation accuracies, and since machine learning models trained on data sets that exhibit great standard variation typically accumulate bias, color normalization could potentially be a workaround for this and actually improve the applicability of deep learning models in a clinical context. And so some limitations of this research or this study was the fact that it only used one data set. That data set had a limited size, and also only one deep learning architecture was used for testing. Also, although very popular and conventionalized color normalization techniques were tested, there are still so many more that were not tested. And a future direction to carry on this research would be to obtain more data and to use more models for testing. And something specific in color normalization that I'm interested in is to see the potential of generative AI, specifically generative adversarial networks, to act as color normalization techniques, and how those could impact the landscape. And yeah, I want to thank you guys for listening.